So I guess I'll start with this notion of genomics as a tool. And the question that I often hear from ecologists and evolutionary biologists is, why do we need genomics? Why do we need to get at the gene? Is genomics a millstone or is it a milestone? And Dick Lewontin was asked this question 25 years ago when he was asked to reflect on the start of the molecular evolution revolution in the late 60s. And he was asked this question, and he said, you know what, it's a millstone. He considered this notion of electrophoresis and the onset of this massive genetic data to contribute to what he referred to as the considerable depauperization of the diversity of empirical work in evolutionary genetics. So I guess he was a little bit grumpy, perhaps like James. James still has some time. If we were to ask him 25 years from now to reflect on the onset of NGS, what would we say? But part of this was, interestingly, for somebody like Dick Lewontin, he attributed this to the discovery of the joys of electrophoresis. Now, I don't know about you, but Mason, as he just qualified in his own work in the lab, there's a lot of blood, sweat, and tears that go along with both electrophoresis and next generation sequencing data. So I guess, from this, I think that what Dick Lewontin was really getting at was this need for integration of various levels in evolution and ecology to fully benefit from this genomics revolution, from this molecular evolution revolution. And in 2003, there was the first Gordon Research Conference on Ecological Genomics. Back then it was called Ecological and Evolutionary Functional Genomics. But collectively we decided that was far too long for an acronym. And so traditionally we just consider it to be ecological genomics now. And the general goal of ecological genomics is to determine what are the proximate and ultimate causes of evolutionary change. And to get at this, we are really talking about a causal link between genotype, phenotype, and the environment. Now, in most cases, it can be a little tricky to determine what is the link between the phenotype and the environment. In some cases, it's a little easier. James wondered if I'd have a bird in my talk, and indeed, this is an example of perhaps where there's not a good fit between the phenotype and the environment here. I didn't realize they gave Darwin Awards to animals, but apparently this bird won one. So in terms of why this is important, why do we need to find a causal link between gene, phenotype, and the environment? Well, it's not only in terms of determining that fit between form and function. Why are organisms so well suited to their environment? It's also because that same process, natural selection, the driving force of all of this, is also associated with the origin of species. And increasingly, we're appreciating that most species seem to originate by means of natural selection. So it's also important in the context of speciation. Now, the framework for ecological genomics essentially takes one of two approaches. You can either start at the phenotype and work your way down to the gene in terms of determining the function. Is this particular gene under selection? Or you can work your way from the gene back up to the phenotype. In either case, there are incredible challenges associated with each one of these avenues going from phenotype down to genotype. And ultimately, in order to be able to find that link with the environment, you have to be able to determine whether or not there's effect of fitness in association with the genes and their environment. And the problem, or one of the problems, is that environments vary. So Dobzhansky would remind us that when we're studying these things and when we're studying how traits are inherited, we have to keep in mind that what is inherited is not just a specific character or trait. What's inherited is a definitive norm of reaction to environmental stimuli. Now, Dick Lewontin actually took this one step further, which is maybe not surprising. He was one of Dobzhansky's students. And he said that when you consider that genomes propose and environments dispose, you have to also consider that the environment selects phenotypic variants, but also proposes with the genotype the variant that it then selects. So the environment can work with the gene to give rise to phenotypic variation that can be subject to natural selection in an environment. So, in terms of the questions that I have for today, I'd like to talk about this framework in terms of the ways that the environment can propose with the genotype to give rise to phenotypic variation, but also what are the evolutionary consequences of this? And ultimately, where can some of these tools fit in terms of helping us figure out this process? And I've chosen two areas of research in our lab right now. I'm going to talk about phenotypic plasticity, so the ability of a gene to give rise to environmentally sensitive phenotypes, so phenotypes that differ in environments, but also phenotypic integration, so the notion that traits covary. And I've chosen to talk about this in the context of fishes, which is one of the main research focuses that we have in our lab. Okay, so the story starts in terms of the history of the region that we study. So in Canada, as most of you know, during the Pleistocene was completely covered in ice. So it was 1 kilometer thick sheet of ice, obviously very heavy, considered to be one of the most significant events in the distribution and abundance of extant species today. About 12,000 years ago, we started to see the recession of that ice, the rebound of areas, especially in coastal regions. And we like to call this essentially evolution's repeated experiment, because all over this landscape, we had the formation of postglacial lakes that were areas of tremendous ecological opportunity. And they were colonized. And in the context of fishes, we have 242 approximately freshwater species of fishes in Canada. And remarkably, in many of these environments, we've been able to witness very rapid evolution, very rapid adaptive divergence that has given rise to species pairs in a number of different species. Now, the species that I want to focus on today is the threespine stickleback. When I was putting this presentation together, I found an old photo of the Schluter and Peichel labs. I don't know why we did this, but we actually had, I guess you could call it a gang sign of threespine stickleback. So if you really want to represent with the stickleback, you can try that next time. I had to do it. So the threespine stickleback is found in marine environments, coastal lagoons, and repeatedly in these postglacial lakes it has colonized fresh water, and we see bouts of adaptive divergence. So here I've got a marine threespine stickleback, freshwater threespine stickleback. We can see one of the major traits to show rapid adaptive divergence in association with these lateral plates, which act, we think, as a defense in the marine environment. And in freshwater, these are completely reduced, along with other bony features such as the spines, reduction of the pelvic girdle, and overall a transition in terms of a suite of shape characters in association with the freshwater environment. We see this parallel evolution occur in hundreds of lakes, giving rise to the same changes over and over. And in the context of the genetic basis of adaptation, genes of large effect have been found for pelvic girdle, lateral plates, pigmentation, and a number of genes associated with phenotypic plasticity. Even the nature of peak shift in these freshwater environments has been shown to be consistent with the predictions of Fisher-Orr geometric model of adaptation in terms of the genetic effects and the distribution effects that occur in freshwater environments. So it provides a highly tractable way to test some of these questions, and in terms of the collection of itself, if you have a bucket and a minnow trap, you can find yourself some stickleback. Now, in terms of its emergence as a model vertebrate organism, yes, we do have a reference genome. It is quite nice. And this is thanks to the hard work of Felicity Jones and David Kingsley and his group, which did include the Broad Institute at Stanford University. And our genome is approximately 463 million base pairs. Here we've got showing each of the linkage groups. And here you can see, in terms of these peaks along the linkage groups, this is where they've actually gone and sequenced a number of marine and freshwater stickleback. So there was a question about population genetics. This has been at least started in this species. So they've got genomes from 10 marine, 10 freshwater. That's it, though. And in that context, when they can compare those genomes, we start to see regions associated with molecular divergence between environments. So these would be considered to be regions of the genomes that are potentially good candidates to be associated with adaptive divergence. Now, in terms of the genetics of adaptation, it's very useful in the context that we can bring these species into a common environment, we can grow them in the lab, we can hatch them in a beer cup, and because of that and the tremendous diversity we can see, it allows us to really get at the inheritance of a number of traits of interest. So when I started this work a long time ago, and I'm talking like Mason back in the day, the blood, sweat, and tears, we had, I think, 7 microsatellite loci at our disposal. Now we have competing genome browsers, so it really is changing face in terms of being able to proceed to the stickleback browser. Now, the first question that I want to talk about today is in regards to the influence of phenotypic plasticity. We all know that phenotypic plasticity is the ability of a gene to give rise to different phenotypes in different environmental conditions. And in terms of some of the 2 main predictions of phenotypic plasticity, my student Matthew Morris, a PhD student in the lab, was interested in 2 primary questions in association with the influence of plasticity on adaptive divergence. The first was associated with the prediction that plasticity should facilitate population persistence following colonization of a new environment. So the genomic norms of reaction that you see following colonization of a new environment would facilitate the ability of populations to persist in these changing or new environments. The second prediction is in association with the evolution of plasticity. And following colonization of a heterogeneous environment, there's a nice prediction that plasticity should actually evolve in association with this new environment. So Matthew set out to test this in association with temperature changes between marine and freshwater environments. Where we work in the Pacific Northwest, we find that, generally speaking, the freshwaters are much more variable seasonally in association with temperature. So we tend to see lower lows and higher highs in association with these 2 environments. So we also know that in the context of the evolution of temperature-related adaptive changes, an experiment that I had done during my postdoc, where we took marine fish and we colonized freshwater ponds, we found that thermal tolerance, in particular the ability to tolerate cold temperatures, evolved at a very rapid pace. So here we actually quantified this in the context of haldanes, and we can see that the ability of fish to evolve and tolerance to cold temperatures was almost about 1 standard deviation per generation. So that's very rapid evolution in this new environment. But one of the criticisms that we did get in association with this, well, what is the influence of plasticity in this context? And so this is what Matthew set out to test. So the way that Matthew did this is he used the same marine and freshwater populations that we had used in that study, and he used F1 full SIB from replicate populations, so we had 2 of each type. He tagged and randomly assigned these F1s to 2 temperature treatments, 7 degrees and 22 degrees, meant to mimic or closely mimic the upper and lower thresholds of temperature tolerance in this species based on their critical thermal minimum and maximum temperature. And then he allowed these fish to persist in these environments for 1,700 degree days. In terms of what phenotype to look at, Matthew decided to use one of the tools that had become available as a result of the genome sequence that was present at the time. And Erica Leader, one of the collaborators on this project, had actually developed a 60,000 gene species-specific array. There's approximately 20,000 genes in the three-spine stickleback, but on the microarray, they're actually replicated 3 times each, so this gives us some technical replication to allow us to measure gene expression. So gene expression, these 20,000 genes, in many ways can be considered a molecular phenotype. It's the abundance of mRNA transcript that's being produced in association with these 2 environments. So Matthew extracted RNA from the muscle. He then used these microarrays to hybridize the RNA onto the microarray, measure gene expression, and test these 2 questions associated with population persistence and orthoplasy. So in terms of population persistence, what Matthew found was that gene expression plasticity existed in association with over 5,700 genes in the stickleback genome. So these are genes that were similarly plastic, similarly exhibited genomic norms of reaction in association with temperature in both environments. So these are the most likely candidates to be associated with population persistence in the face of environmental change. In terms of the second question, was evolution implicated following colonization of freshwater environments? If so, we predicted that we should see more plastic genes implicated in freshwater populations compared to the marine ancestors. And in fact, that's what he found. He found significantly more genes with plastic expression than marine populations. So following this, as with most transcriptomic studies, we have a series of genes that show some enrichment. Indeed, a lot of these genes were associated with temperature or energy metabolism. But we decided again to benefit from one of the tools that we have at our disposal, which is the stickleback genome. So we went in and we actually looked which of these candidates, in particular, the ones that had evolved in association with freshwater, were also under selection globally between marine and freshwater populations. And what Matthew found when he compared this with the genome sequence of the three-spine stickleback, there was one gene in particular, PPARA, there's no easy way to say it. I wish that we had better names for genes. That's my wish list. That's where I'm going to get grumpy. Hypothetical protein number 1 just doesn't cut it. PPARA, so peroxisome proliferator activated receptor alpha A. We know that this particular gene, interestingly enough, is associated with transcriptional regulation. In this case, transcriptional regulation of mitochondria biogenesis, amongst other things. So what Matthew found is a significant reaction norm in association with freshwater where this gene is upregulated in freshwater. When we look on the stickleback genome, on linkage group 4, just downstream from our old friend ectodisplasin, which is the gene associated with lateral plates, when I say just downstream, it's about 7 million base pairs downstream from that. We find a nice signature of selection between marine and freshwater populations. And these SNPs, this particular gene, is within this region of SNPs that show this outlier level of divergence. So Matthew decided to take this one step further. He wasn't happy with the phenotype of gene expression for this gene that we were looking at, and he wanted to study what this might actually mean in terms of an adaptive response to temperature. And so what he did was make the prediction, the very simple prediction from his previous findings, that given this, marine stickleback should not exhibit increased mitochondrial density in the marines. You'll see it in the freshwater, but not in the marines at low temperatures. And the way that he decided to test this was to conduct another experiment where he reared stickleback for marine and freshwater environments for 9 weeks, this time at 6 and 20 degrees. And following this, he harvested pectoral and cardiac muscle, and I now find myself doing electron microscopy. So again, just another level of phenotype. And we don't have the exact results yet, But what we do have are some very recent pictures that for somebody that does this sort of thing, the technician that helped us wasn't excited at all by this. Yeah, there's your mitochondria. We can see them here in the pectoral muscle, and we can see them here in the cardiac muscle. And so what Matthew will be able to do is take this one step further and look at phenotypic variation in association with the density of mitochondria and also the surface area to fully test this prediction. Following that, if this holds, then it becomes compelling to potentially look further for the mutation that might be associated with explaining this mechanistic basis of phenotypic plasticity. So I'm going to switch gears a little bit and talk just briefly about phenotypic integration. This is work being done by Tegan Berry, who is currently a master's student starting a PhD in the lab. And Tegan's always been interested in the notion that organisms are integrated to function as a whole. So when we talk about phenotypes, we really need to be talking about phenotypes in the context of covariation and how quantitative traits may covary. We know that this tends to be either because of correlated selection or for genetic reasons such as pleiotropy or gametic phase disequilibrium. And this is why, again, it's important to eventually incorporate genomic tools into this sort of question. One of the things that we would predict is that if variation is modular and it's evolving in response to selection, this should favor integration of phenotypes following colonization of new environments. So Tegan has set forward doing this using another new tool that we find ourselves using in the lab to characterize phenotypes, and that's computed tomography, CT scans. And the focus right now is on what we refer to as the trophic apparatus. So this is the skull of the fish. And so when we talk about morphological integration, what we're really talking about is covariation between different aspects of the skull. So for instance, if we consider these regions of this trophic apparatus, so the opercular region, the skull or upper skull region, the jaw, if we find that two parts of the phenotype display weak covariation but are strong internally, then we would say the phenotype is modular. If we find that two parts of the phenotype display strong covariation but are weak internally, this is when we can say that the phenotype is integrated. So with respect to stickleback adaptation to freshwater, one of the predictions that we would make based on this is that regardless of the modules that we're looking at within this trophic apparatus, we would predict they would be similar regardless of population, because they are integrated to function as a whole. We've decided to test this on a variety of modules that are present in the skull, but today I'm just going to talk about what we refer to as the opercular 4-bar lever. So this is a series of 4 bones that start primarily with the operculum. So this is where I get to pretend to be a fish. This is the gill covering of the fish. And the other bones extend to the jaw region. So generally speaking, the opercular 4-bar lever functions in terms of respiration, but also feeding. It so happens when we disrupt this module for whatever reason, we reduce the efficiency of predation and respiration kinematics. And this has been shown experimentally. We can use what's called the RV coefficient. So essentially, we can measure covariation between sets of landmarks to ask these questions about phenotypic integration in marine and freshwater environments. And so I'm just going to talk a little bit about some data that we just published. I'm not going to talk about all the modules, but talking about the opercular 4-bar lever, we do see significant differences in terms of the phenotype between marine and freshwater fishes. You can see obvious variation in terms of other aspects of the skull. This falls out nicely in terms of PC plot between marine and freshwater populations. Now, interestingly, an F2 cross, a hybrid between these two sources, does fall intermediate. That's what we would expect in terms of genetic variation raised in a common environment. But when we look at phenotypic integration in the opercular 4-bar lever, we find that it is significant in terms of being integrated in marine and in freshwater, but not in the F2s. So it's disrupted in the F2s. And right now, we're not clear whether this disruption is a functional mismatch in the phenotype, but we have found in some intermediate natural populations between marine and freshwater, we see the same sort of thing. There seems to be functional mismatch in terms of these modules. So this is setting the stage for some experiments to follow up on the effects of fitness that this might have, but also in terms of seeing whether patterns of phenotypic variation and covariation match with genetic variation, and covariation is where Tegan will be going with this. And recent work in our lab, QTL mapping the trophic apparatus from these 3D CT scans, has shown, importantly, that next generation sequencing is going to be an important component of this to get down to the regions that are underlying this phenotypic variation. But importantly, the 3D CT scans are also showing us that the phenotype is important. We detect better and more QTL using these 3D CT scans than we do with conventional two-dimensional techniques. Now, from this, we also have students moving back into nature to test some of these other questions that we have, in particular the three-spine stickleback genome and those 21 fish representing marine and freshwater populations didn't look at regions of hybridization that you find intermediate to those populations. So these are the regions where we're finding some of these functional mismatches. So Ella Bowles, a PhD student in the lab, has been sampling stickleback in a very interesting setting in Katmai National Park, Alaska, where we find three-spine stickleback marine and freshwaters existing in symmetry, but also in allopatry with marine and freshwater counterparts. And Stevie van der Zwaan, who apparently looks ready for the grizzly bears that like to step on our traps up there, has been actually looking at covariation and phenotypes in these environments. And so I won't go too much into this because they have a poster here that I encourage you to look at, but using about 8,000 SNPs from similar method that Janneke talked about this morning, the dd-RADseq, we are finding evidence that indeed this is part of the reason why these aren't included in full genome sequences, because we are finding evidence of admixture between marines and freshwaters. In addition to that, when we look at the phenotypic variation, we're finding tremendous phenotypic variation in association with these freshwater environments, even in cases where we're finding very little genetic differentiation between populations. So with that, I think that going back to being grumpy a little bit, again, I think there is the potential for next generation to be a millstone. I think where it has the real potential to be a milestone is when we consider the importance of the phenotype, which is the variation that selection actually sees. From this context, I am increasingly becoming appreciative of the notion that environments do vary, and this variation can actually contribute to phenotypic variation in ways that I think need to be considered in the context of adaptive divergence. And I've given you two examples where this seems to be influencing evolution of three-spine stickleback. And so to finalize, a couple of years ago, we celebrated 20th anniversary of molecular ecology, and John Slate, Aurelie Bonin, and Louis Bernatchez and I were asked to write about the priorities in ecological genomics for a special issue in that journal. And with respect to ecological genomics, it's becoming increasingly clear that we really do need theory in association with these empirical studies. We need extended evolutionary theory that can start to generate some discussion from the theoretical side when we talk about evolution, development, quantitative genetics, ecology. Along those lines, we really do need more ecological annotation of genes. A lot of the genes that we work with need functional ecological annotation. And in that context, phenotypes are not going away. We need to be very proactive about measuring phenotypes. And along the lines of this talk, hopefully we're going to be more predictive about solutions that organisms have to environmental variation and heterogeneity. And in this context, experiments are going to be continued to be important. So with that, again, I'd like to thank Jana for the invite. Especially like to thank my lab. This is a very dynamic, wonderful group of people that continually challenge me. So it's a very tremendous learning environment right now. And with that, the various collaborators that work with us on this project. Heather ran the Boston Marathon yesterday, so let's hope she did all right. Thank you very much.
Well, there's lots of time to talk to Sean over lunch, which, oh, we do have a question? Oh, sorry.
Thank you. No. Hi.
Hi, Abby.
I'm just teasing because I'm very curious about all of this. But, you know, we were at a biogeography conference actually, and I don't know, we got off on a tangent, and I asked Louis Bernatchez about some of these populations where you had initially the contiguous habitat, so it could have been the St. Lawrence River or, you know, in BC, where you had this the saltwater reversal and sort of how far saltwater intruded into sort of the freshwater environment. And I'm just curious, so this is really interesting, and I wonder if any of your students or if you are looking at the physiology, so in terms of salt tolerance or...
So we haven't, in terms of physiology, absolutely. I mean, I think a lot of the studies in association with temperature, it really comes back to energetics and physiology. And so I didn't talk about, I focused more on the mitochondrial side, but again, that's associated with the delivery of oxygen, energy. So I think that that's where that definitely relates to that. In terms of salinity, that just reinforces the nature of environmental heterogeneity, and we have work on another species, prickly sculpin in the Fraser River Valley, where we've used pool seek, which is just a slightly different version of getting at SNPs into environments. And the prickly sculpin, similar to threespine stickleback, extends along this gradient of marine to freshwater, and genes like sodium, NAK, ATPase, associated with salinity tolerance definitely pop out. So I think that reinforces the need, even when we talk about covariation between morphological traits, there's definitely influences, or almost certainly influences, of covariation when we consider things like salinity tolerance and bone formation, for example. So all of these things eventually need to be considered in that context of adaptive divergence to new environments. Yeah, it's a great question.
Thank you.